Moreover, the effect of temperature on amylase activity can be investigated using a different approach, which may also be presented in exams. Let's explore an alternative method for carrying out this experiment. This experiment can also be carried out in a different way. In this method, the investigation began by heating a starch solution to a specific temperature, such as 35 degrees Celsius. A thermometer was used throughout to ensure the temperature remained steady. At the same time, a spotting tile was prepared by adding a few drops of iodine solution into each well. To confirm that the iodine was working correctly, a drop of starch solution was placed into one of the wells. As expected, it turned blue-black, indicating the presence of starch. Once the starch solution reached the desired temperature, the amylase enzyme was added to initiate the reaction, and the timer was started immediately. At regular intervals, about once every minute, a small sample of the reaction mixture was transferred into a new well on the iodine tile. The color change was closely observed. As amylase began to break down the starch, the blue-black color gradually faded. When the sample no longer turned blue-black and stayed yellow-brown, it indicated that all the starch had been broken down. The time taken to reach this stage was recorded, representing the rate of enzyme activity. This procedure was repeated at various temperatures, such as 20 degrees Celsius, 40 degrees Celsius, and 60 degrees Celsius, to study how temperature affects the activity of amylase. Results typically showed that amylase functions most efficiently at an optimal temperature, often around 40 degrees Celsius, while activity decreases significantly at temperatures above or below this point. This method clearly demonstrated the effect of temperature on enzyme activity. So, this is how temperature affects the time it takes for amylase to break down starch. As you can see here, the process is shown in 3 main stages. First, we heat the solutions. We place 2 test tubes in a beaker of water that's been set to the temperature we want to test. This could be 20, 35, 40, or 60 degrees Celsius, for example. One test tube contains 1 cc of amylase, which is a yellow liquid. The other holds 5 cc of starch solution, which is a reddish-orange color. Next, we mix the solutions. Once both have reached the desired temperature, we pour them into the same container, usually a beaker or a conical flask. This starts the reaction. The starch and amylase are now combined, and we begin timing immediately. Finally, we test the mixture for starch. To do this, we use a spotting tile with drops of iodine already in each well. Every 30 seconds, we take a small sample from the reaction mixture and add it to a new well on the tile. If starch is still present, the iodine will turn blue-black. As the starch is broken down by the amylase, that blue-black color fades. When the mixture no longer causes a color change, when it stays yellow-brown, that tells us all the starch has been digested. We record the time it takes to reach that point, and by repeating the experiment at different temperatures, we can see how temperature affects the activity of the enzyme.